Hi everyone, this is Lauren Schmitz. I'm going to jump right in. This is joint work with James Lee, Kyung-Shi Liu, and Jason Fletcher. We're all at UW-Madison. So we have two aims for this project. The first is to expand on past work that's evaluated the joint effect of tobacco control policies and genetic risk factors on smoking and related comorbidities. We're going to be using multiple population cohorts and data from a large multi-center RCT. And we're also seeking to improve how we're modeling G by creating polygenic scores that leverage genomic and epigenomic functional annotation. And our hypothesis is that these polygenic scores that prioritize cell-specific pathways may improve the detection of G by E interactions and the clinical utility of G by E studies. And I'm really going to focus on the second bullet point in this short presentation. So as it turns out, the majority of GWAS hits are in non-coding DNA. And many of these SNPs are actually in regions of regulatory DNA. What is regulatory DNA? It's any region of DNA that regulates transcription. So why does this matter for G by E? Well, dynamic regions that regulate transcription are the most sensitive to environmental forces. However, standard GWAS weights are capturing the average effect of a SNP on a phenotype across all cellular states and types. And so the effect sizes of regulatory variants may be understated, making it more difficult to detect a G by E signal. Whole genome aggregation also masks specific genomic regions in cellular programs that are interacting with the environment. And so this makes it more difficult to identify potential behavioral or pharmacotherapy interventions. So the question is, can we construct a polygenic score that is tissue-specific and allows us to investigate the impact of these really important regulatory regions? And the answer is yes. My amazing colleague here at UW-Madison, Kyung-Shi Liu, has developed a method called AnnoPred that leverages genomic and epigenomic annotation to adjust GWAS signals. So this slide is showing G by E p-values from separate models that use different polygenic scores interacted with state-level cigarette taxes in adolescence. And the outcome here is smoking initiation. The PGS on the far left is the smoking initiation polygenic score using weights from GScan, all SNPs, no clumping or pruning. And then in the middle, we have an AnnoPred smoking initiation polygenic score that utilizes genomic and epigenomic functional annotation across the genome to adjust GWAS signals. We're still using the GScan weights here. And you can see that this really significantly improves the p-value on the interaction. And finally, on the far right, we're using AnnoPred smoking initiation scores and functional annotation that's specific to regions of the central nervous system in the brain. And you can see that this actually performs best. So next steps, we feel that these results are really exciting, but they are preliminary. So we'd like to continue testing AnnoPred polygenic scores that upweight other areas that are important for smoking behavior, including the adrenal pancreas and cardiovascular systems. And we'd also like to use functional annotation related specifically to enhancer and promoter regions. These are really important regulatory regions on the genome. We're going to carry out replication analyses in AdHealth and Midas. And we're also going to use data from the lung health study, which is a large RCT, to look at the effects of behavioral interventions and genetics on cessation and cardiovascular outcomes, also using AnnoPred scores. We're currently in the process of resubmitting an R21 to support this work, and I'd like to acknowledge additional collaborators on this submission, Lauren Gaydosh and Sean David. So thank you, and look forward to seeing you at the poster.